Hello, welcome to another tutorial from the Biology for You. In this video, we are going to discuss Roche 454 DNA sequencing technique. In this series of videos, we have been discussing different methods in DNA sequencing. So, this Roche 454 DNA sequencing is basically one of the very popular method from next generation DNA sequencing. So, these next generation DNA sequencing techniques are basically high throughput methods which can sequence very large DNAs like genomic DNA or maybe DNA from the chromosomes from the higher organisms. So like that, this with the help of next generation DNA sequencing, we can sequence very large DNA in a single stretch. So these next generation DNA sequencing techniques are high throughput methods in which the DNAs will be sequenced in massive and parallelized manner. Sequencing will happen. So, in the next generation DNA sequencing, basically the principles may be different, but there are some common steps like the large DNA will be fragmented, or that will be cleaved with the help of restriction enzymes to form short fragments which can be easily processed. And from all these fragments, the clone will be prepared with the PCR amplification. Very large set of copies are prepared from every fragment. So likewise, for each fragment there will be a clone which containing some very large number of copies, and for each copy we are going to sequence the DNA. So there will be a very large sequences obtained for a single fragment. So likewise, for every fragment there will be a clone and all will be sequenced. So the efficiency of the sequencing will be increased. Efficiency is high. Apart from the efficiency, generally all these process carried in a single stretch of experiment. That means here the PCR amplification of the all fragments will be carried and simultaneously, and also the sequencing of the all fragments will be carried simultaneously. So that means it decreases the time. So these next generation DNA sequencing techniques are very much speed and they are very much efficient as well as, and apart from these characters, these next generation DNA sequencing techniques will sequence the DNA in a single stretch. The whole genomic DNA will be sequenced in a single stretch. That means they speed up the process of sequencing. And also, the popular examples from the next generation DNA sequencing techniques are Illumina technique, Illumina DNA sequencing, Roche 454 DNA sequencing, and the ion torrent. So in this video we are going to have the basic concept of Roche 454 DNA sequencing technique. So coming to that point, these are, as we already discussed, these are type of next generation DNA sequencing technologies. And in the Roche 454 DNA sequencing, here the basic steps are like, once after you isolate the DNA, you need to go for emulsion PCR. Emulsion PCR to amplify the DNA fragments. And next is pyrosequencing. So the basic sequencing procedure is the pyrosequencing. So this is the large form of pyrosequencing. And here, so before going to the main PCR amplification, we also need to do library preparation. So during this library preparation, which is the first step in this Roche 454 DNA sequencing, here we need to let us consider it as a large DNA that is going to be sequenced. Here we need to make fragmentation. So the large DNA that is going to be sequenced will be treated with the restriction enzymes, which forms the shorter fragments of the double-stranded DNA. And the fragment length, the average fragment length in this Roche 454 is some 300 to 800 base pairs. So in the next step, we are going to add 3' and 5' adapters to every sequence. So for all fragments obtained from the genomic DNA, we are going to have two kinds of adapters. One is 3' adapter and other is 5' adapter. So in the next step, we need to go for denaturation. So maybe the alkaline treatment will be given to break the hydrogen bonds present between the two strands of each fragment to obtain single-stranded DNAs with two adapters attached on both of their ends. So that is what the single-stranded DNA, ssDNA library. So all these single-stranded fragments together called as the library obtained from the genomic DNA that is going to be sequenced. So that is the first step. So next two steps are the key steps from this Roche 454 DNA sequencing, that means emulsion PCR. Here this emulsion PCR, in which these single-stranded DNA attached with these two adapters will be attached with the bead. So all fragments obtained from the library will be attached with these beads. So here in emulsion PCR, these beads are flooded or emulsified with these PCR essentials. That means in the emulsion there will be water and oil mixture. So this water and oil droplet will be coated around these beads. So all beads will be coated with this water and oil microreactor. So this droplet, simple droplet, is called as microreactor, in which the bead is provided with the PCR essentials. So the basic importance of this emulsion PCR or maybe the microreactor is to provide space for individual PCR reactions. So here the bead is provided with the PCR essentials present in the microreactor, that is emulsion that is coated around the bead, and this provides this space to amplify into very large set of copies. So in a single bead there may be chance of the producing up to million copies of the DNAs from a single template. So likewise, with the help of emulsion PCR, by the end of this PCR amplification, we are going to have million copies of fragment, single-stranded copies from each template. That means each single-stranded fragment obtained from the genomic DNA library. So all this process is simultaneous for every fragment. So by the end of PCR PCR technique, the all fragments obtained from the genomic DNA are amplified into some millions of copies. So that is the emulsion PCR's importance. So in the following step, these beads are allowed for the sequencing. So once after this PCR amplification is carried, these PCR products, that means the bead coated with the single strands, are allowed to be sequenced. So here all these copies that are amplified will be again attached with the same bead on which the template is present. So here all these fragments in the sequencing procedure are allowed to synthesize their complementary strand. So during the synthesis of complementary strand, we are going to sequence the DNA of it. That means the addition of nucleotide will be monitored at every nucleotide as an addition during the complementary strand synthesis. So this is basically sequencing based on synthesis procedure. So in the pyrosequencing, the basic technique is like that. So let us consider there is a nucleotide addition during the synthesis process, the complementary strand synthesis. Here this nucleotide is going to be added. So due to phosphodiester bond formation with the adjacent nucleotide which is already existed in this sequence, there will be release of pyrophosphate. That means among the three phosphate groups, 3 phosphates, 2 will be released and 1 will be involved in the phosphodiester formation, bond formation. So the release of pyrophosphate here going to be monitored. So here we'll get again one question, that is how we'll understand what type of nucleotide is added when the pyrophosphate is released, because for all types of nucleotides the pyrophosphate is the same. So during pyrosequencing, first we need to understand what type of nucleotide is going to be added. So for that, generally here in this pyrosequencing procedure, we will not add all types of nucleotides, all type of DNTPs at once. But instead of adding all types of nucleotides at once, we are going to give these nucleotides in a flood, in a wave fashion. That means a type of nucleotide, say for example DATP, will be added to the synthesis PCR amplification during the PCR amplification first. Then we will be monitoring in whether it is producing signal or not. So if it is producing a signal, that means at the first nucleotide position there is a DATP going to that is added on the sequence. So if there is no signal, we can consider there is no DATP in that position, first nucleotide position. Likewise, once after this DATP is added, we are going to add DGTP, then DCTP, then DTP. So likewise, in a wave fashion, one by one, each type of nucleotide is added, and we need to check whether the signal is produced or not. If signal is produced, that means at that nucleotide position there is that type of nucleotide particularly. So likewise the pyrosequencing will be carried, and during this procedure, pyrosequencing procedure, generally sequencing procedure, all these emulsion PCR products, that means these beads containing single stranded of copies, will be added onto the flow cell or the sequencer plate. containing number of nanopores on it. So the size of nanopore is such that it only accommodates a single bead. So the single bead containing the DNA, number of DNA copies will be added to each nanopore. So each nanopore represents the fragment obtained from the genomic DNA library. So genomic DNA or maybe genomic DNA. So here the PCR products are allowed to sequence by their synthesis of the complementary strands. Here we are going to add all PCR essentials like enzyme, primer and all except the DATP or DNTPs. So once after all these PCR essentials are added, these DNTPs will be added in a wave fashion. That means DATP added in a single step, followed by DZTP will be added, and DCTP will be added in the following step. Like that, one by one, these nucleotides will be added, and once after a type of nucleotide is added on the sequencer plate, we need to check in which nanopores or the pores the signal is produced. That means if the signal is produced, the light is produced from the first nanopore or the first well present on the sequencer plate, that means here is the addition of DATP, and if there is no signal in the second well, we can consider it as there is no DATP in that position. Likewise, for every nucleotide addition, we need to check what all are the wells that are giving signal and which all are the wells that are not producing signal. So detector system will be there, and computer program will be there which can monitor continuously all these signals, and the wells which are not producing signals, they will be recorded simultaneously. So likewise, till the end of the sequence, the signal will be recorded. So how do they produce light? That means how do they produce signal for every individual addition of the nucleotide? That is what we need to discuss here again. So for that, as we already discussed, addition of a nucleotide will produce signal. The release of pyrophosphate, and the release of pyrophosphate is a signal. So how do the release of pyrophosphate will provide signal? So there is again an enzymatic reaction. That means the release of pyrophosphate, the released pyrophosphate will be reacts with, there is an enzyme called sulforylase which add APS to the released pyrophosphate. So this APS is nothing but the adenosine phosphosulfate. Here this APS will be added to the each pyrophosphate that is released in the each nucleotide addition event, and here ATP is produced, and in the second step with the enzyme called luciferase. This is the second enzyme. This luciferase is generally obtained from the firefly, which produce the light in natural system. So this enzyme is used here, this firefly's enzyme is used here, and there is a compound called luciferin in it. So the released ATP will oxidize this luciferin, present in the luciferase enzyme. So with the help of ATP, luciferin is oxidized into oxyluciferin and AMP will be produced. That means energy is consumed up and CO2 is the byproduct, and again a photon is released. So this photon is the main reason for the production of the light whenever the pyrophosphate is released. So that is how the light will be produced, or the signal will be produced at every nucleotide addition. So that is how the pyrosequencing will be carried. So up to here, in this Roche 454 DNA sequencing technology, we discussed how do we process the DNA isolated for the sequencing. That means here under library preparation we fragmented the DNA, and every fragment is denatured, and there is an adopter ligation. So these single stranded DNAs with two adopters attached on their both ends are the products from the first step. And during the emulsion PCR, the beads are attached with every single strand of the DNA fragment, and these beads are emulsified with PCR essentials, so that PCR amplification is carried in this microreactor, and there will be very large number of copies obtained from every piece once after this PCR amplification is carried. So during the pyrosequencing, here the beads are loaded onto the sequencer plate containing nanopores. So here every fragment, every clone that is obtained from the fragment from the genomic DNA is sequenced simultaneously in the sequencer plate in every well. So here we are going to add these PCR essentials at a time, but the DNTPs are provided at last in a waves fashion. That means every type of nucleotide will be added one by one. So that means at a type of nucleotide added at once, followed by the other type of nucleotide. So once after this type, a single type of nucleotide is being added, we will be checking for the wells which give the light signal, and that signals will be recorded. So here, like that, till the end of the DNA synthesis, we are going to have this sequence. So which wells are producing signal, which wells are not producing signal, that will be detected by the detector system. So that is how the Roche 454 DNA sequencing will be carried.